Check. Okay. Yeah. Yeah. So welcome back. So there is an issue with the voice. So these are the articles that we are going to cover for today. West Nile fever, that is a part of science and technology, right? Recently some cases were recorded in Kerala. Then CRISPR-Cas9 tool, again part of science and technology. So those students who are preparing for those exams which are having science and technology environment as a huge weightage should prepare thoroughly, because we are covering lot of science and technology and as well as environmental articles every day, not just today. Then Sahyadri Tiger Reserve, part of environment. Then trade related aspects of intellectual property rights. So TRIPS agreement as part of economics. Then leopard census, again part of environment. So it was already discussed, but again it was there in news, we will see about that as well. Then we have 4-5 factual pointers and finally practice questions. So welcome to the session, a very good evening to everyone. Yeah, so we will start with the first article for today, that is West Nile fever. So the context is why we are taking this article is that the Kerala has reported a death as well as some more cases related to the West Nile fever recently, a few days back, right. So that's why there was an article in the Hindu. So let's see what is actually West Nile fever. So West Nile is a region, Nile is a region in Africa, right. So there is a Nile river. So this was origin in the African continent, we will see that as well in which year it was reported. Before that, let's see what actually this type of virus or type of disease is. So West Nile fever is caused by virus called as West Nile virus. These names of viruses are given usually, diseases or names of virus are given usually from the regions where it was first discovered, right. So this is a virus and this is a single stranded virus. So we have a double helical DNA, isn't it? So we have DNA like this for humans, we have a double helical structure, and certain virus has only single, that is called as RNA, ribonucleic acid, right, and humans and other mammals have DNA, which is a double helical structure, right. So this virus is a single structure, that is it is having a RNA, not DNA, right, and this belongs to the flavivirus. These facts are important. There are number of subgroups within mosquitoes, you no need to worry about it, arbovirus or adenovirus, there are certain things, but for this West Nile virus you need to remember this. This is belong to the flavivirus of the mosquitoes, right, and this was caused by Culex mosquito. Culex mosquito. So there are again types of mosquitoes where Aedes aegypti, so we have number of such type of mosquitoes. So this is caused by the Culex mosquito, right, and this same flavivirus can cause the Japanese encephalitis, right, and yellow virus or yellow fever. So this flavivirus may also, the same family of virus may also cause this type of disease. So this West Nile fever is related to the virus and it is caused by the single stranded virus, right. So this is commonly seen in Africa, Europe, Asia, Middle East region and North America commonly. Of course all continents has been covered, so it is not just specific to Africa, but it is prevalent across the world, that is what we want to tell, right. And this usually peaks, the number of cases of West Nile fever usually peaks where the mosquitoes breeding season is higher, and mosquitoes breed in either in rainy season with the higher temperatures. Whenever there is a cool temperature, the mosquitoes cannot be breed, right. So this is the conditions. Now how can it be spread, right, why it is causing? So if you observe this, these are the hosts, here this means birds. So birds are the hosts of this virus, right. So we have birds as the host, that means the virus is within birds, and then when mosquitoes bite these birds, then the virus in the bird's blood is carried to the mosquitoes, right. So birds are the hosts and this what you call mosquitoes are the carriers, right, they are trying to spread. They are sucking this blood from birds and then they are biting on animals or humans. Then it may cause this West Nile virus. Is this clear? So, birds are the hosts and mosquitoes are sucking this blood and then they are spreading to the humans as well as the animals, right. So, this is how the diseases are spread, and this disease will not be able to spread from humans to humans. There is no such case recorded. Only when mosquitoes spread, when mosquitoes take this virus from this birds and then they carry to the humans, right. So, there is no direct contact. If you are standing beside any person who is having West Nile fevers, no, it will not spread. But if the blood of that person is taken and then it is given to some other person, yes, it may cause. So, it is not contagious. It is not a disease which can spread direct through contact, but rather it can contact or it can spread through blood transfusion or through the mosquitoes from the birds, right. So, this is the spread of this particular disease. Now, as I said, it was caused by Culex mosquito. This is a very important one. So, it is not known to spread with the direct contact with animals or humans until unless there is a blood transfusion. So, this is the transmission method of West Nile fever, and there are a few more features about this disease or this is virus you have to know. This may cause neurological disease. This may cause this virus if sustained in the human. So, it may cause neurological disorders, right. So, they will not be able to function their brain effectively, or it may also cause the death, right. So, this fact is also important, and this was first isolated from a woman from West Nile in Uganda. This fact is also important. So, it was first discovered from Uganda country in 1937, and yes, in India approximately all the states has reported some cases of West Nile virus, right. And 80% of this West Nile virus fever is what you call asymptomatic. That means we cannot see the symptoms. Usually it have symptoms of fever, headache, vomitings, rashes and all, but 80% of the people will not show the symptoms itself. So, that is also very important to be remembered, and as of now there is no specific treatment or vaccine for this disease. Still there is a work going on, but there is no vaccine or medication was discovered till now, right. So, this is about the West Nile fever. So, the most important thing is you have to know the context and then where they are being spread, what is the features of this disease and how it is being spread, who are acting as a host, who are acting as a carriers, and who can it spread and what way they can spread, right. So, this is about the West Nile fever. Any doubts in this? Any doubts? So, very simple article, right. So, now let us take the second article. If you have any doubts, please let me know about this particular article that we have discussed, or else we will move to the next article, that is CRISPR-Cas9 tool. CRISPR-Cas9 tool, a very important, many times been asked in the exam, either in conventional, that is answer writing as well as objective exams. So, yeah, great. So, the next article is CRISPR-Cas9, and why we are taking this article? This was taken from the Hindu, that it has reported a person who is facing, few persons who were facing lack of vision, right, they have some sight issues. So, this was able to cure even the disease. CRISPR-Cas9 was like genetically we have known for some other diseases, but even eyesight can be able to rectified using the CRISPR-Cas9. So, we will see first about CRISPR-Cas9, how it works and all, then we will go to the actual what type of disease it was cured, right. So, let us understand CRISPR-Cas9. You all know that our human body is having cells, number of cells, millions of cells, and within cell we have nucleus, right. Within nucleus we have chromosome, within chromosome we have DNA, right. This is the mechanism: cell, then nucleus, then chromosomes. How many set of chromosomes are there? Anyone? How many set of chromosomes are present in human body? Chromosomes. So, we have 23 set, that means 46, and then we have inside DNA, right. So, this is the structure of the cell. Now, there will be certain, as we have seen inside the chromosomes there is a DNA, and some DNAs may be defective because of various reasons of birth or may be inherited. There may be some issues with our DNA, and that issues may cause some diseases, right. So there are certain what you call genetically developed disease, right. So like Down syndrome, we have many other things, genetical disease. Now the scientists across the world has come up with a new tool called as CRISPR-Cas9, clustered regular interspaced short palindrome repeats tool, CRISPR tool, right. Cas9 is a protein, we will see about that as well. So this CRISPR-Cas9 is having, CRISPR-Cas9 is a tool which can edit the DNA. We have seen that certain part of DNAs can be defected and those DNAs can be edited. We will see the process, do not worry. Just understand that it is a tool, it is a technology which can edit our inside DNA, right. So inside DNA, so that the defected one will be taken and the proper one will can be attached. So this is the mechanism, or this is the definition of CRISPR technology. Now within CRISPR we have two important components. The CRISPR-Cas9 system consists of two important components. One is an enzyme called Cas9. So this is a protein or enzyme, Cas9 enzyme. This acts as a scissor. So if you want to cut any thread, what you are using? You are using the thread scissor. So this Cas9 is like a scissor which the protein can attach it to the DNA, and wherever there it is selected, right, wherever the in whichever DNA it want to edit, so it can attach and it can cut. So this is the enzyme which can cut the DNA, the defected DNA. So this is one component, and the second component is the guide RNA. So for example, this is a DNA, right. So this is DNA and you want to cut this portion, right, or you want to cut this portion. So what is single stranded DNA is called, so I mean the genetic material is called single, it is called as RNA, right. So DNA is a double stranded and RNA is a single strand. Now Cas9 we have seen, Cas9 we have seen it is an enzyme which can attach and it can cut this portion. And the next component is guide RNA. It is nothing but when you are going to cut this RNA is going to attach the proper one, because we do not want double helical structure, we just want a single strand, right. So this is a RNA, guide RNA, which after Cas9 cuts this portion, this RNA will attach to it and now it will become a proper RNA. And this RNA is designed like based on the persons, based on the requirement they are designed, and it goes and attached to the particular place which we cut in our DNA. So now it become a functional one, right. So this is the two components. Now if you see the diagram, yes, right, 23 pairs are there in our human body. So now understand the process, this is a very important thing. If you do not understand the process how it works, and if you by heart, you will forget in the exam. So the most important thing is you need to remember the mechanism, how it is working. So your brain should always be curious on something, else it is very difficult to clear these exams, right. Now it is more about analytical skills than remembering the things, right. So if you see the technique, this is our cell, and within nucleus we have a chromosomes, and within chromosome we have a DNA. So this is what we have seen. Now Cas9 is an enzyme, and we have seen that this red color one is a defected one. So this single strand is a defected one in our human body, right, for any person. Now this Cas9 will attach till here and it will break, right. So this will be cut in our DNA, and then this guided RNA, so guided molecule or guided RNA then will be attached. So after Cas9 gets cut here, because only single stranded one was cut, right. So if for example this part and this part is dysfunctional, still we need a RNA this side and this side, right, we need a two single strands to attach it. So here it is it was cut by the Cas9, and then this green color one is the guided RNA which can attach. So there is a mechanisms and all to attach the RNA. So you no need to worry about going into the research, that is not necessary. Just understand that it is going to attach and then it work as a regular DNA. So, this is the process of using CRISPR-Cas9 technology, right. Is this clear? Is this clear, anyone? Please do respond if you are not clear with it. Is this clear what we have discussed about DNA editing, that is CRISPR-Cas9 technology? Or should I repeat it again? So, this is about, we have seen the cell and then the existing one, to cut any part, the defected one is cut using the Cas9, and then the guided RNA is attached to the DNA, the cut DNA, and then it becomes as a regular functional one, right. So, this is the gene editing technology. Great. Now, let us go ahead a little bit more what the scientists has discovered. We have seen that scientists were able to cure the eyesight as well, right, eyesight as well. So, there is a disease called Leber congenital amaurosis. So, this is a lack of eyesight because of genetical defection. So, in such persons there is a genetical dysfunction, and this causes the Leber congenital amaurosis. So, like any other genetical disease, in the eye also there is a genetical disease called as LCA. So, for this purposes, the scientist has used CRISPR-Cas9 technology called as, this was given some other name, yes, EDIT 101. It is nothing but a new name given by the own scientist, because they have done the new thing. So, they have given this CRISPR-Cas9 technology a new name, EDIT 101, right. So, this CRISPR-Cas9 technology was used, and the mission, the trial name was given as Brilliance. They may ask you these things also in the exam. So, recently the term Brilliance project was seen in news, what is that, something like that they will ask. So, this is a trial to conduct on 14 persons, 2 children and then 12 actual persons who are facing this disease, Leber congenital amaurosis disease, right. So, they have deployed EDIT 101, which is nothing but a CRISPR-Cas9 technology, and then they have tested it, right. After that, it was proved that there was a mutation, I mean the DNA, there is a different DNAs, different names are given. So, here CEP290 was a DNA, defuncted DNA. So, the Cas9 has cut this one, and then they have replaced with a guide RNA with a proper mechanism. So, what it was resulted, it proved that most of the people out of 14 were able to see the vision properly, right. So, they were able to see whatever the things that were not able to see in before, now they were able to see it clearly. So, it means that it want to say in future you no need to get specs, or you no need to go for any LASIK treatments and all, if it is having genetical things. So, they can edit and then they can make you to have a proper vision. So, this is how what it has happened. Malini, the one which we are cutting is called as using Cas9. Cas9 is the one which is using an enzyme which can cut the DNA, right, which can cut the DNA. Now, the one which we are attaching, that is called as guide RNA. Guide RNA. So, as I said, DNA is like this, right, double stranded DNA is like this, but only one thread is cut, like in whatever portion it is defected, it is cut using Cas9, and then this guide RNA, a single one, you can see in this diagram, this green color, yeah, this one will be replaced, the green color one. So, it is cut and then it is attached to this. This is called as guide RNA, because it is a single strand, right, which we are attaching. Is this clear? Please do let me know if you still have any doubts. Is this clear, Malini? I hope this is clear. Now, the next article, that is Sahyadri Tiger Reserve. Sahyadri Tiger Reserve. The context is, there is a Sahyadri Tiger Reserve, but in that there is no proper tigers. Though it is a tiger reserve, we do not have tigers in that. So, that is why Maharashtra government is going to shift the tigers from some other tiger reserve, that is Tadoba Andheri Tiger Reserve. This is also located in the Maharashtra itself. So, within Maharashtra from one tiger reserve to other tiger reserve they are going to shift, relocate some tigers, right. Yeah, great. So, they are going to relocate those tigers from one tiger reserve, that is Tadoba Andheri Tiger Reserve, to the Sahyadri Tiger Reserve, because there is a lack of tigers in Sahyadri Tiger Reserve. So, for our exam point of view, we need to know Sahyadri Tiger Reserve, we need to know Tadoba Andheri Tiger Reserve, and from which tiger reserve to which tiger reserve they are shifting the tigers, and where they are located, both are located in Maharashtra. So, these facts are very, very important. Now, let us understand Sahyadri Tiger Reserve. First understand the geography, first observe the geography where they are located. So, Sahyadri Tiger Reserve is located in the Western Ghats, right, Western Maharashtra, and this is the only tiger reserve in the Western Maharashtra, right, in the Western Maharashtra. So, other tiger reserves you can see Tadoba Andheri Tiger Reserve, which is border to Maharashtra and Telangana. So, from here they are going to shift some tigers towards this region, right. So, this is the objective, and we have some other tiger reserves in Maharashtra, you can see here as well. This is Telangana, right. So, Kaval as well as Amrabad tiger reserves are located in Telangana, but anyway they are going to shift the tigers from Tadoba Andheri to Sahyadri Tiger Reserve. So, that is the objective. So, here Tadoba Sahyadri Tiger Reserve is located in the Western Ghats. Important thing is they are located in the Sahyadri ranges and then Western Ghats as well. And this tiger reserve is formed from the areas of two important sanctuary as well as national park, Koya Wildlife Sanctuary. So, tiger reserve means a tiger reserve declared separately for the protection of tigers, and this tiger reserve may have some 2-3 more wildlife sanctuaries, national parks and all, right. So, Koina Wildlife Sanctuary, which is located again in Maharashtra, and then Chandoli National Park. So, from these two regions together they have formed the tiger reserve. They will ask you this also, that Sahyadri Tiger Reserve consists of which of the following wildlife sanctuaries or national park, right, Koina and then Chandoli. So, that is important, that need to be remembered. And the habitat of Sahyadri is known for Sada. So, it is a type of plant. So, this is popular. So, that also you need to remember. So, it is a latter, a type of plant in the Sahyadri ranges, right. Now, we also need to know Tadoba Andheri Tiger Reserve as well. So, Tadoba is a name of a god. So, a local god in Maharashtra region of western or the southern Maharashtra region, and Andheri is a river. Andheri is a river. So, because this tiger reserve was constructed near this Tadoba, a popular god where the people worship, and then Andheri river which is passing through. So, together it was called as Tadoba Andheri Tiger Reserve, right. So, that is also important, and this have national park also. This tiger reserve has certain part of Tadoba National Park and Andheri Wildlife Sanctuary. So, this also they may ask you in the exam. These are facts you need to remember. Tadoba National Park, Maharashtra, Andheri Wildlife Sanctuary, Maharashtra, certain part of these two were together formed as a tiger reserve, alright. So, this you need to remember. Now, what is the program about? We are coming back to the context that they are relocating. So, the thing is they want to translocate few tigers from Tadoba Andheri Tiger Reserve towards the Sahyadri Tiger Reserve, right, to spread, because in some national parks there are huge amount of tigers and the habitat is not able to sustain, because the area is small and tigers need a freedom to roam. So, that is why they are relocating. Those higher number of tigers are there in Tadoba and then they are shifting to the Sahyadri, and the long term plan is to revive the northern Western Ghats. Is a huge amount of area which can give lot of habitat for tigers, but tiger number is not very huge in northern Western Ghats. So, that is why the main aim is to shift some tigers from different regions towards the northern Western Ghats, and this Sahyadri Tiger Reserve, which government is going to shift some tigers, can act as a corridor. So, this Western Ghats, whole Western Ghats can act as a corridor, and why we need corridor? Because tigers cannot, they can stay longer for certain region. They want a longer one to roam, like how we want to roam to different places. tigers also want to roam. So this is the Western Ghats, and ultimately government is aiming to create a corridor for tigers to roam, to have a larger area. So in the future they are going to integrate all these wildlife sanctuaries. You can remember Radhanagiri Wildlife Sanctuary, again Maharashtra, Amboli Reserve Forest in Maharashtra, Mahadev Wildlife Sanctuary in Goa, Bhimgad Wildlife Sanctuary and Kali Tiger Reserve in Karnataka. So they are going to construct a integrated corridor without any disturbances. Tigers can roam around all these tiger reserves as well as wildlife sanctuaries. So that is the aim, right? So in the initial phase they are going to test by shifting some tigers, and later they are going to also shift from Pench Tiger Reserve to other parts, these tiger reserves as well, right? So that is about the project. Any doubts? So in future they are going to shift from Pench Tiger Reserves, 8 tigers they may shift, right? So next article, trade related aspects of intellectual property rights. First let us understand what are intellectual property rights. Anyone? Can anyone answer what actually is a intellectual property rights? Intellectual property rights. So intellectual property rights are those ideas derived from our mind. For example, I have developed one vaccine and someone might have copied it and they might commercialize. So the idea actually is mine, but someone is benefiting out of it. So intellectual property rights are those rights on your ideas which may enable the creation of goods and services. So those are also materialistic. So if you are having a good idea, if you are making a good idea to produce some good, so it may helpful to most of the people, and you can you can materialize it because of your idea. The crores business has developed, right? So those are intellectual property rights. Now the same, earlier the world is isolated. One country was situated at one region and they are not able to integrate with the other countries, but now the world is integrated. After like 30-40 years, before itself the world has integrated. Our thoughts may transfer to other country as well. That's why an agreement was signed under World Trade Organization, that someone who have created in one country cannot be copied by other country members until unless you pay something and take permission, right? So TRIPS, trade related aspects of intellectual property rights. Our ideas should not be copied even in other countries also. So this is the agreement, right? Now the context is, it was completed 30, this is the 30th anniversary of signing this trade related intellectual property rights agreement, right? So let us understand some facts about it. It was signed under WTO, World Trade Organization, in 1995. In 1995, very very important. It was negotiated in 1995, and what it guarantees? It guarantees minimum standards for IP protection, intellectual property rights. So you have to register and publish in certain database so that all the countries will also know. Even if some person is copied and if it is not known to others, so this database will help to identify that this person was the initial one who has invented these thoughts, right? So he has to be given, or she has to be given certain money if you want to copy it, right? So this is the mechanism, but still the agreement provisions give certain challenge. For example, I have discovered one one idea which is helpful to the lot of people, like COVID vaccine, and if I says that I need to be paid with 1000 crores, but the world is not ready because they don't have money. So in such health emergencies, even without his or her permission, who has developed, the countries can use. So this TRIPS agreement is enabling the countries to bypass. There was a provision that during the health emergencies, not in every case, but during the emergencies wherever the world is require this idea, then this is not applicable. So you cannot claim any money. So government, even if that person don't want to buy, or sell, this worlds can use those things and then they can produce the goods for the larger benefit of the society, right? So that is the TRIPS provisions, right? It is protecting the intellectual property rights. Along with that, it is also giving certain comforts or grace to the world nations when there is a requirement for the humans, so you can use it, right. So that is why most of the countries have signed, and India is also part of this agreement, right. This is about the trade intellectual property rights. Yeah, patents is a process. So patent is a process where you file it, right. Intellectual properties are ideas and patents are the registration. Whatever the registration you do, that I have invented this, and they will give in turn the certificate. Those certificates are patents, right. So that is about the trade related intellectual property rights. I hope this is clear. Now let us move to the last article today, that is International Leopard Day. International Leopard Day. So we will also see the leopard census, but yeah, on May 3rd we have celebrated the International Leopard Day. That is why we have taken this article from Indian Express explained page. Before knowing the database of leopards in India, let us understand the facts related to the leopard as well. So leopard in India is found in almost all type of forest. It is not just about tropical, temperate or northeast, it is almost everywhere found in India. There is no restriction as such, right. And International Day, as we said, it was celebrated on May 3rd every year. This is listed as vulnerable in IUCN list, very important, right. They are listed as vulnerable. So we have seen IUCN in many times the categorization, starting from data deficit, least concern, near threatened, right, vulnerable, endangered, critically endangered, extinct in wild and extinct. So we have seen this. So it is protected under vulnerable category and under CITES agreement, right. So CITES is an agreement internationally to protect those species which can migrate, which can be transferred from one country to other country, right. So this is protected with the higher status, and under Wildlife Protection Act it was under the Schedule 1, which is having the highest protection status, right. Now let us see the 5th cycle of leopard population, which has done recently by the government, right. So these numbers are also very important. The total leopard population was estimated to be around 13,874 approximately, but it is not accurate. So the range is between this, and they have estimated to be 13,874 leopards in our India. So that is one important thing, and this was increased compared to the last census. So there is an increase, there is no decline, there is a increase. So it is a positive element, and this was estimated across the regions. We have Himalayas, we have Peninsula, we have Eastern Ghats, Western Ghats, right. We have different regions, and in except Himalayas, okay, the Himalayas parts of the country which are not tiger habitat were not sampled. Yeah. So here the Himalayan ranges does not have leopards. We have snow leopards, we have tigers as well, but Himalayan and semi arid regions we do not have usually high number of leopards. Very rarely we can sight these leopards. So that is why these regions are not surveyed. So from the surveyed regions of other regions like Western Ghats, Eastern Ghats and all, the central India shows a stable growing population. The central India is showing a stable, including the Eastern Ghats, Western Ghats, central plateau, right, peninsular plateau. This is having a higher numbers. The number has increased, but there was a decline in two regions that you need to remember: Shivalik Hills and then Gangetic Plains. Ganga Plains and Shivalik Hills is the southernmost hills, right, near Himachal Pradesh, Uttarakhand, the southern side of Himalayas. So these two regions have seen the decline, but other regions, central India and then Eastern Ghats, Western Ghats, it has seen the increase in the leopard population. And among the states, Madhya Pradesh is having the highest leopard population with 3,907, followed by Madhya Pradesh and then Karnataka. Top 3 you have to remember: Madhya Pradesh, then Maharashtra and then Karnataka. So these 3 are the top 3 states with the leopard population. And the tiger reserves, among the tiger reserves which are having higher number, it is Nagarjun Sagar Srisailam Tiger Reserve. This is the largest tiger reserve in the Yeah, so there was a interruption, sorry for this. So we have seen till here. So Nagarjun, among the tiger reserves that we are having, Nagarjun Sagar Srisailam Tiger Reserve is having the highest number of tigers. So this was the largest tiger reserve in India. This was asked in UPSC 2020 prelims, UPSC prelims. So this is the largest tiger reserve, and the highest numbers of leopards are located in the Nagarjun Sagar Srisailam Tiger Reserve, which is located in Andhra Pradesh, right? And followed by Panna Tiger Reserve and Satpura Tiger Reserve. So these are the top 3 where within Tiger Reserve we have a higher number of leopards, right? So these are the fact, and we have seen already that Shivalik Hills and Gangetic Plains were the regions where there is a decline in number, right? So this also you need to remember, right? Shivaliks and Gangetic. In other regions, Western Ghats, Eastern Ghats and other northeast, we have seen a higher numbers, increase in number, right? So this is about the leopard numbers. And now who has conducted this data, I mean who has conducted this survey? This is also important. This was conducted by National Tiger Conservation Authority, which is responsibility, which was created under Wildlife Protection Act. Wildlife Protection Act, it is a statutory body, right? It was created to take care of the tiger reserves. The same organization was associated with the conducting of the survey, right? Along with the support of Wildlife Institute of India and then state forest departments. So these 3, like state forest departments, NTCA, and then Wildlife Institute of India, these 3 together have conducted this, and this is part of, for every 4 years they are conducting these estimates, and this is the, after 2019 we got this report, right? So these numbers are very, very important. So we have seen about the context, the leopard day on May 3rd. So we have seen the conservation status and other details of leopard, and then we have seen the numbers which was recently released by the government, right? So which states are having highest, where they have increased, where there was a decline, and which tiger reserves are hosting more number of leopards. All these facts are very, very important, and who has conducted this survey is also important, right? Any doubts in this? Right. So now comes the factual pointers. Recently there was a press day, we have seen also, recently International Press Day we have celebrated. So in the Indian Express has published a report on Bengal Gazette. So Bengal Gazette is a first newspaper in India. Bengal Gazette is a first newspaper in India, and this was very important for India. It was launched in 1780 by James August Hickey. Yeah, James Augustus Hickey, an Ireland person, who this was the first newspaper in India, right? So it was a weekly English language paper. It is not a Bengali or other language, Hindi language. So it is an English language newspaper, right? So this is also important, and it was published in the Kolkata region. All the facts related to it is very, very important. Who has published it? Even though the name is Bengal Gazette, it is not in Bengali, but it is published in English, and that too is a weekly paper, weekly ones, right? It was published in Calcutta, and this was closed down after 2 years because this paper has criticized then Governor General Warren Hastings. So Warren Hastings was our the Governor General that time, first Governor General. So this magazine has criticized that person, so that's why it was closed in 1782, right? Next one, about MQ-9B. They will ask you what is this MQ-9B. It is nothing but a drone. It is like a drone, but it seems to be a helicopter or plane, but it is a drone, predatory drone. That means whatever the enemies, if it considered, it will blast. Of course the accuracy is based on software and all, so any enemies if they see, so it can target, right? So this is a drone, MQ-9B. This is going to be deployed by Indian Army and Indian Air Force. Next fact, Department of Consumer Affairs, this was taken from PIB. There is a concept called as drip pricing, drip pricing. So Department of Consumer Affairs warned the businesses that if you are publishing drip pricing, then it is a crime, civil crime, so we will take action. And now what is actually a drip pricing? So if you see any advertisement, the hotels, for example, you have visited some hotels and it was showing price as 1999, but actually if you go to the hotel after, like when you booked and when you go to the hotel, it may be 2599. So what they have showed is different. Now they will add other things like GST or some Wi-Fi, internet, swimming pool, whatever may be, and they are charging it as a higher number, higher amount. So drip pricing is a instant or a process where they show, the companies show the lesser amount, but actually when you are consuming it directly, the prices are higher. So those are called as drip pricing. So Department of Consumer Affairs has warned that if you want to show, you show it 2599 itself. Don't show the reduced price and attract them. Once they book and once they go there, they may charge higher. So that should not be happen, it will be penalized, right? So this is called as the drip pricing. I hope this is clear. Right. So the last fact, recently Kutch Ajrak, it is an embroidery work, you can see here it is an embroidery work. Kutch Ajrak has got the GI tag from Gujarat. So it got a GI tag. GI tag means any locality is having speciality in producing that, right? So certain regions in certain states, certain districts have certain speciality, right? So those are given with geotag that this is the region where it was emerged and it has a speciality, right? So this was given for Kutch Ajrak, fine. So these are the factual pointers for today. Now let's practice some questions from the topics that we have discussed, right? First question, with reference to West Nile virus, which of the following statements are correct? The Aedes species of mosquito act as a principal vector for the transmission. First statement. Second statement, the disease is asymptomatic in 80% of the infected people. So that is the second statement. It is highly contagious through human to human contact. This is the third statement. So what is the answer for this? Anyone? We have discussed this West Nile virus, right? First article itself. So is this Aedes species of mosquito? No, it is of Culex mosquito. So first can be eliminated. The disease is asymptomatic in 80% of infected people. Yeah, we have seen only 20% people are shown these symptoms, 80% are asymptomatic. And the third, it is highly contagious through human to human contact. No, this is also wrong, right? So this is not contagious. Contagious means whenever you go direct to the person who was have this disease, so it will spread to the other person. No, it is spread through blood transfusion. Blood transfusion has to happen, or mosquito has shift from some blood from other things, virus, to other person. So answer is B. Ranjan Malini Oendila has answered it as correct. Very good. Next question. Which of the following statements regarding CRISPR technology is correct? CRISPR technology is correct. CRISPR technology does not involve the introduction of any new gene from the outside. Any new gene from outside. See, this is one point I want to emphasize. When we were discussing, I think we forgot it. So in CRISPR-Cas9, the RNA, right, the guide RNA which we are attaching, it is nothing but from our own genes itself. We are not taking some other genes, some other persons and all. We are generating it artificially and then we are attaching it. So we are not taking any outside material, right? So it is our human body genes itself. So as the context come, I am saying it. So this is the first statement. Single guided RNA is one of the components of CRISPR. A special protein called Cas9 is used to break the DNA strand at the specific points and remove the bad sequence. So tell me the answer. So, first statement, it does not involve the introduction of any new gene from outside. I just discussed this point that we are not involving any new genes, it is our own human body, right, our own human body. For example, we are having this DNA, right, and this is affected, right. So, what we are doing, we are taking some other DNA, the same, we are studying this particular string, and then we are making it same way outside in the laboratory, and then we are attaching it, that is guided RNA. So, we are not taking outside of gene, it is our own gene itself, we are developing and then we are giving it, right. So, first statement is correct, second statement is also correct, we are one of the components, and the Cas9 is also enzyme which can remove. So, answer is D 1, 2 and 3, yes, correct. So, Ranjan has answered it as correct. Next question, which of the following defines the term drip pricing? Drip pricing. So, we have seen this also. Let us see who will answer this question. It is a commercial pricing strategy which involves the use of large scale undercutting to eliminate competition, right. So, to undercutting means suddenly you are reducing prices and then occupying all the customers, then you are increasing the prices after eliminating all the competitors in the market. So, this is first definition they have given. It is a pricing technique in which firms advertise only part of the product's price, So, that is the second statement. It is a process of conveying a false impression or misleading information about how companies' products are environmentally sound. So, this is the third statement. A misquote where the price quoted material are clearly deviate from the prevailing underlying market prices. So, what is the answer for this? First one is predatory pricing. See, what is predatory pricing? When some company like Jio, they reduce the cost of all the internet and all the people will rush there to buy, and then will increase the prices. That is predatory pricing. But we are talking about the drip pricing, which means that pricing techniques in which firms advise only part of it and rest of the part they will price it when you are consuming it, right. So, the answer is B, yes. Srinath Malini Oyindila has answered it as correct, great. Next one, Sahyadri Tiger Reserve is located in. So, Sahyadri Tiger Reserve is located in which state? Madhya Pradesh, Maharashtra, Odisha, Goa. So, it is not Goa, it is not Odisha. So, you may confuse between these two. This is located in the Maharashtra region, western Maharashtra, isn't it? So, it is a very direct question. Right, correct. So, answer is B, yes. Last question, which of the following tiger reserve or national park holds the highest leopards in India? Sahyadri Tiger Reserve, Nagarjuna Sagar Tiger Reserve, Kali Tiger Reserve, Satpura Tiger Reserve. So, what is the answer for this? We have seen the largest tiger reserve or national park, Nagarjuna Sagar, which is having the highest number, right. So, answer is B, yes, correct. Yeah, so that is of today's session. There is a lot of facts, of course, you have to remember, understand the CRISPR and rest of the things you have to by heart, right. So, this is of today's session. Thanks for your patience who are staying in the sessions for longer time, and but really it will be helpful. Have a great evening and happy studies. Thank you.